Hey everyone, how's it going? I'm here to talk to you a little bit more about protein synthesis. So I provided you with a little practice sheet, and that's not going to be turned in for an assignment or anything, but it's a chance for you to practice some of the things we've been talking about and hopefully help you understand this idea a little bit more. It's an important idea. So, if you haven't, I encourage you, go ahead and go and work through those practice problems first. And then in this lecture, we'll talk about them, okay? And so, of course, we're talking about protein synthesis. We're talking about DNA, which makes RNA, which makes protein. And again, I want to help you understand why I make such a big deal out of this. So first off, traditionally, this is kind of the least favorite part of the class whenever I've asked students before, and usually, students kind of struggle with this part a little bit. And it's like, why do we make such a big deal about it? Well, because it is a big deal. Like, you just can't understand biology unless you have a good grasp of this. And it's the thing that unites all living organisms. Again, from bacterium to a fungus to a tree to a bald eagle. They all do it, and they all do it in the same way. And that's fascinating. And understanding this process really helps you understand lots of phenomena in biology. When we're unlocking the secrets of the DNA. That's where we're at in 21st century biology. And so, to be up to speed, you have to understand this a little bit. I understand that we're just taking a superficial view. There's a whole lot of details we're leaving out here. But what I'm hoping to get is I'm hoping for you to get, a basic understanding and have a good enough idea of how all this process works, this process of protein synthesis works. And then that's going to help us understand a lot of other things that we're going to talk about. So, for example, things that are better explained now that we understand protein synthesis. Things like a lot of genetic disorders. And so these are disorders that have been identified, and then as they're studied more, you start to understand what's happening at the molecular level, and you can understand and hopefully treat some of these disorders. And so, for example, a mutation in a gene is going to change the RNA, and it sometimes changes the protein. And changing an important protein might be detrimental to the organism, and that might cause a genetic disorder. So a good example of this, there's lots of these, but a good example is cystic fibrosis. And so this is a genetic disorder, and it's characterized by a lot of things, and again, there's a lot of details we're leaving out here, but one of the characteristics of cystic fibrosis is thick mucus in the lungs. And so this becomes a problem because that thick mucus harbors bacteria and viruses and makes the functioning of the lungs more difficult. And that could be a problem. With thinner mucus, it can flow and you can cough it out, and that's one of the things we talked about. Part of your immune system is things like mucus that traps particles before they get into your body. But then you need to be able to expectorate them. You need to be able to get rid of them. But if you got this thick mucus, you can't, and things like that. Now, there's several different mutations that have been identified and several different forms of this gene that causes cystic fibrosis. But a very common mutation leads to the loss of a single amino acid out of about almost 1,500. So you've got this protein that's about 1,500 amino acids long, and there's a mutation that leaves out a single amino acid. And this is in a protein called the CTFR protein. And leaving out that amino acid changes the shape of this protein enough that it doesn't work properly. And so that's how a mutation can lead to a genetic disorder. And so here, here's an example. What does this protein do? So this kind of calls back to some stuff we've been talking about in class. Remember, we talked about the cell membrane And that's what you're looking at here. You see the phospholipid bilayer, right? Every cell is surrounded by this phospholipid bilayer. And we said there's lots of proteins on the surface and going through the cell membrane. And so the CTFR protein is a protein that goes all the way through the membrane. It sits in the membrane, but it makes a channel in the membrane. It allows stuff to flow through that channel. And so this is like poking a hole in the membrane. Remember, we talked about the big deal about the cell membrane. It's semi-permeable. It lets some stuff in, it lets some stuff out. And one of the ways that the membrane controls what goes in and out is through having channels. And stuff can only cross the membrane through these channels, and so then you can better control what goes in and out. And those channels are most often big proteins. And this is an example. This CTFR protein makes channels in the cell membranes. And it's found in places like the lungs and maybe the intestine and other places, right? And so essentially what this channel does is it allows ions out, mostly chloride ions. So you've got a lot of chloride ions inside your cell, and you've got a low concentration outside. Well, think back to what we learned about diffusion. Where are those chloride ions going to go? They're going to diffuse from high concentration to low concentration. And so they want to diffuse out of the cell. And this CTFR protein allows them to diffuse out of the cell. Those ions can't just go through the membrane, but they can go through this channel created by this protein. And so because of diffusion, those ions diffuse out of the cell. Well then, now you've got more solutes outside the cell. You've got things outside the cell, it becomes slightly hypertonic. And you remember what we said about osmosis and the diffusion of water. Water always flows to the hypertonic location. So you've got more stuff dissolved outside the cell now, and the water is going to follow. Now, in real life, there's lots of this going on all the time with all your cells, and remember we talked about homeostasis. That's an important part of homeostasis, is maintaining the salt-water balance. But this is one way that it happens. And so, ultimately, how is this related to what we're talking about? Well, if this is functioning properly and the water and the ions are allowed to flow out of the cell, that gets water outside the cell, and that's where the mucus is. And so then it thins the mucus to a consistency that your body wants, and that's how a normally functioning CTFR protein works. But in this particular mutation we're talking about, that protein is missing an amino acid that changes its shape just a little bit. And so the protein is still there, but its channel is blocked. So just that one little change caused the shape to change enough that now it doesn't work as a channel, or at least it doesn't work as well as it should. So the ions can't flow out of the cell, which means water is not going to follow. Water is not going to flow out of the cell, which means you're not getting water to that mucus. And so that mucus gets thick and difficult to expel, and it harbors bacteria and all those sort of things. And so my point here is, this is how understanding, how proteins are made and what they're made of and how they relate to DNA, now you can understand a biological phenomenon like cystic fibrosis, and then hopefully you can start to figure out ways to treat it. And so now there are new drugs that are designed to help this protein hold its shape, or to help hold the channel open. But the point is that, until you understand how this protein is made and what's causing it to misfunction, you can't really come up with a drug. But once you understand how it works, now you can start to understand drugs and treatments and things. And so that's where we're at. That's where a lot of a growth in our knowledge is coming now. And so this is an example of why understanding protein synthesis is so important. Another example. And again, this is pulling back and relating to things we've already talked about. Remember that most genes have several alleles, right? Different forms of that gene. And so that allows different organisms to have different genotypes. So different organisms have different combinations of those alleles, and that's a genotype. And that often leads to a different phenotype. And so here's another example. And so there's a mouse that lives down in Florida called the Oldfield mouse, Paramyscus pollionotus. Yeah, pollionotus. And so this is just a mouse that just lives down in Florida. And these mice have a gene called MC1R. And that's just the name of the gene. And it's just, again, a part of a chromosome. What does this gene do? This gene produces, all genes do. They produce RNA, they produce protein. In this example, the protein that this MC1R gene makes is an enzyme. Again, you remember what an enzyme is. An enzyme helps chemical reactions go faster. And the reaction that this enzyme catalyzes makes a dark brown pigment. And so to make pigment for the fur, you need some sort of chemical reaction. You need to synthesize this pigment, and that's a chemical reaction. And this enzyme helps that to happen, all right? And so this gene gives these mice a brown color. At some point in their evolutionary past, a mutation occurred, and it changed a single amino acid in this enzyme. So again, in the last example, we deleted an amino acid. Now we're just changing. We're substituting an amino acid. So you've got this long string of amino acids. The DNA changes, the RNA changes, and we substitute. We have a new amino acid in one spot. All right? Well, that changed the shape of the enzyme. The enzyme doesn't work as well as the original enzyme, and so it doesn't produce pigment as efficiently. And so mice that have this mutated gene don't produce as much pigment as those that don't have the mutated gene. And so here's some pictures of these different kind of mice, and it's adult mice, and you can also see how the coloration actually is established, at birth, because on the right-hand side you've got young baby mice. And so if you've got a functioning MC1R gene, or 1R allele, that produces pigment, and it gives the mouse a dark brown pigment on top. But if a mouse has the mutated MC1R allele, that doesn't produce pigment as efficiently, and so they're not as dark. Okay, well, would you call this a genetic disorder? This mutation created a new allele, which created a new phenotype. It created a new trait. Is that a good thing or a bad thing? And so that's where we have to be careful, because words like good and bad, well, they're all relative, and it creates a different trait, but how do we know if it's good or bad? Well, if you look at where these mice are found, the dark brown mice can hide better, they're better camouflaged on the mainland, away from the coast where there's dark soil, and that's what they're showing you. They're showing you the typical soil where this mouse is found. And so if you've got that functioning allele, then you can produce the pigment and you can hide better. And if you've got that mutation, well, now you're a much lighter mouse. You can't hide as well in the mainland, but if you've ever been to the panhandle of Florida, what are the beaches like? White sand. And so if you have this mutated allele, but you live on the beach, if you live nearer the coast, then you're better camouflaged for that environment. And so this mutation created a new protein, which created a new trait, and that actually gave these mice an advantage in a certain place. And so that's another reason why it's so important to understand protein synthesis, is because it leads to new traits, and that's very important for evolution. And so mutations are going to produce new proteins, and that can be good or bad depending upon the environment. And so if you look at this map here, this is sort of a map down of northern Florida, and it shows where you find mice of different colors. Now these are all, the same species, but that you find different colors based upon what environment they're in. If a mouse has that functioning allele and produces dark pigment, but lives on a white beach, it can't hide very well. And so it's going to get eaten very quickly. And so those alleles are going to go away. Whereas the mice that have that new allele, that doesn't produce pigment, they're going to be able to hide better on that white beach, and they're going to stick around, and their babies are going to have the white allele, and that's natural selection. Well, all new traits, every organism is different, and every organism has different traits. All new traits ultimately come from mutations, and that's very important for the evolution of organisms. That son of a bitch. Son of a bitch. Wow. I'd say I got him. Jesus. Take it easy, Mongo. And so that's the point that I'm trying to make, is that knowing the details of how DNA makes RNA and makes protein really helps to give us a deeper understanding of a lot of things in biology. And again, this is, where 21st century biology is right now. So, this is a biology class. It's good for you to know that. So, having said that, that's why I designed that practice. And I want you to kind of work through these problems a little bit so you get a better understanding. So let's talk about it. Let's go ahead and go through the practice a little bit. Okay, so first question. If a gene has a sequence ACG, what's the RNA sequence produced? And so this is kind of the first important idea, right? That these nucleotides always pair up in the same way. So if you know one set of nucleotides, you know the other set. And so if the gene has ACG, then the RNA must be UGC, right? And so, as always, remember that RNA uses uracil, not thymine. And so A always pairs with T. But when we're talking DNA to RNA, A always pairs with U. And then C's always pair with G's. And so if we know that the DNA sequence is ACG, the RNA must be UGC. Okay, so based upon that RNA sequence, what's the amino acid that's going to be inserted into the protein? So that's a codon. And that RNA is going to get read by a ribosome, and that ribosome is going to determine what amino acid to stick in that location. Well, that's based on this chart, this universal genetic code. And so if we look up that codon, which was UGC, you see that it indicates a cysteine. So CYS stands for cysteine. And so that's essentially how this works, right? This code is universal for all living things. So it doesn't matter if this gene is in a fish or a fowl or a blade of grass, that if a ribosome reads UGC on their RNA, it will put a cysteine in to that growing protein that it's building. That's pretty cool. Okay. So now, let's say that original sequence mutated. And so now the gene is ACA. It used to be ACG, now it's ACA. So that third position mutated to an A. So again, what's the new RNA sequence? Well, the RNA must be UGU. So anytime you mutate the DNA, it will also change the RNA. It must. And so now we've got this new codon. So let's go to the chart. What amino acid should go in this position based upon that new codon? Well, it was UGU. And if you look, UGU also puts a cysteine. And so although the DNA mutated, it did not change the amino acid. Remember, there's 20 amino acids, but 64 possible codons. So every amino acid has more than one codon. And so the DNA changed, had a mutation, that changed the RNA, but it did not change the amino acid. This is something that we might call a silent mutation. So the amino acid didn't change, the protein didn't change, even though the DNA changed. So this is an example of how the DNA can mutate and it doesn't affect the organism at all, because the protein won't change. It's the same amino acid. Okay, so did the mutation change the protein? No. The same amino acid was inserted. We don't expect the protein to change, and we don't expect that mutation to affect the organism. That's a silent mutation. So that's one possibility. Let's mutate the DNA again. And so now the original DNA was ACG. Let's pretend it mutated to ACC. And think back to several lectures ago when we talked about what is a mutation, how do mutations happen. There's lots of ways, but every time a cell divides, it has to make copies of the DNA, and sometimes errors are made. In this example, an error is made, and we changed that G to a C. What's the new RNA sequence? It must be UGG. And so now again, if that's the new RNA sequence, what amino acid is going to be produced? Well, if you look, UGG codes for a tryptophan. That's a different amino acid. So there's an example where we mutated the DNA, which of course changed the RNA, and it actually did change the amino acid. Okay? So that's another possibility here. It depends upon where the mutation occurs, and sometimes it causes a change in the amino acid, sometimes it does not, sometimes it deletes an amino acid, sometimes it completely... There's lots of ways the DNA can mutate, but the important thing is to understand is that it may or may not change the protein. And so again, we ask the question, well, in this example, did it change the protein and would it have any effect on the organism? Yes, it did change the protein. It put a different amino acid in that spot. Originally it was supposed to be a cysteine, now it put a tryptophan. So that is a different protein. But we don't know if that's going to affect the organism or not. There's lots of things that could be the result of that change. Sometimes changing an amino acid doesn't change the shape of the protein. It depends upon where the amino acid was and the shape of the protein and lots of complicated... Remember, that protein is a long string of amino acids that all wraps up into a crazy three-dimensional shape. And it might be that, okay, yeah, you could swap out an amino acid at that spot and it doesn't matter. So that happens. But we don't know. You have to look more deeply into the gene. So maybe that didn't change the shape of the protein. But maybe it does. Maybe it does change the shape of the protein, because the sequence of amino acids determines the shape of the protein. So it's possible that swapping out that tryptophan for a cysteine does change the shape of the protein. But again, maybe that's no big deal. Maybe the new shape works just as well as the old shape. That's not common, but it can happen. Maybe the new shape works better than the old shape. It catalyzes the reaction quicker. It's more stable. There's lots of ways that the protein can get better. Maybe the new shape is much worse than the old shape. That happens a lot, right? And so you've got all these amino acids and you've got this protein that's a very complicated shape and it's designed to do one thing. And if you change the shape a little bit, most often that protein stops working well. And so, that's an example of a deleterious mutation. But maybe you've got this new allele and it works good in one environment, but not so well in another environment. Like the example I gave you with the color gene in the mice. And so there's lots of possible things that can happen as a result of this mutation. If you change out an amino acid, there's all kinds of different things. So that's why I say you can't always predict exactly what's going to happen. You can say what amino acid gets swapped out, but you need more details to know if that's going to affect the organism. Okay, so last example, pretend that original DNA mutates, and now the new sequence is ACT. What's the RNA sequence? Well, it would be UGA, right? And so, A's always pair with T's, but RNA does not have a thymine. RNA does not have a T. So when you've got an A in the DNA, that's a U in the RNA. But it doesn't work in the opposite direction. If you've got a T in the DNA, that will still give you an A in the RNA. Right? So don't get confused by that. Just realize that you'll never have a T in RNA. You'll replace it with a U. That's it. That's the only thing you got to worry about. Okay, so what does this new sequence, this new codon, code for? Well, UGA is what we call a stop codon. Remember that the ribosome reads the RNA and reads these codons and then brings in the proper amino acid. And it keeps doing that until it hits a stop codon. And when it hits a stop codon, it goes, okay, I'm done. And it lets go and it assumes the protein is finished. Well, we've just mutated our DNA to create a stop codon where there wasn't one before. That's usually very, very bad, right? Because the ribosome is going to stop making the protein wherever this stop codon is, and that's it. And so there's usually a lot of amino acids that are supposed to come, but they don't get made because this premature stop codon. And so that's another possibility of a mutation, is that you might create just this stub of a protein that's not really going to do anything. So these are all different ways that mutating the DNA can potentially change the protein. And all those examples I gave you are what we call point mutations, a single base change, right? And we saw there's many possible effects, ranging from it's better to there's no change to it's very, very bad, right? Anything can happen from a point mutation. Realize too, there's lots and lots of other ways the DNA can mutate, not just points, but whole sections can get deleted and whole sections can get copied and chromosomes can fuse, and there's all kinds of ways you can change the DNA. We're just getting started. The point is that you can understand what happens with a point mutation. You can see how any other mutation can also have an effect. And like I keep saying in this class, what's interesting is very small changes often have very big effects. And here we're talking about a single base change in the DNA could have very big effects at the organismal level. Okay, so then the last question is, here's a longer piece of DNA. And so I want to see how do you do with a longer piece. So do the same thing that we just did, but take this entire piece of DNA and figure out what's the string of amino acids that come from this part of the gene. And so pause this if you need to do that. Okay, unpause. So let me show you how I do it. The first thing I always do is break it down into codons. Break it down into 3 base pair pieces. It's a lot easier to keep track of what you're doing if you go ahead and break it down into 3 base pair sequences. And so now we've got the DNA divided up into 3 base pair things. How do I know that's DNA? Like if I just gave this to you and didn't tell you anything, how would you know it's DNA? Well, it's got T's in it. If it's got a thymine in there, it's got to be DNA. Now, Also understand that in real life, if it was DNA, there'd be 2 strands here, right? DNA is double-stranded. But when you're transcribing a gene, you only use one of those strands. So let's make sure we keep this all straight in our heads. So I need to make the RNA codons from each of these 3 base pair sequences. And so here are all those RNA codons. And so go through and check yours. But again, A with T, but you can't have a T in the RNA. So if it's going to be, if you've got an A in the DNA, you put a U in the RNA, like this first one, TAC, the proper RNA codon is AUG. C's are always with G's. So these are all the codons from this gene. And so now we can go to the chart and figure out the amino acid for each one of those codons. And so if you look, AUG is methionine. It's also a start codon that does both. Then proline, threonine, leucine, glycine, lysine, alanine. And then your last codon in this example, UAA, codes for a stop. And so there, you've made a short sequence of amino acids. But then in real life, this would be thousands of amino acids, and those amino acids would start to twist and curl, and you would make a very complicated shape, and that would be a functioning protein. Okay, so as always, let me know if you got any questions. I wanted you to get a little practice and maybe solidify this in your brain. But you can see how we're starting to pull a lot of things we've learned. We're starting to pull them together to get a deeper understanding of other biological phenomena. So hopefully we're going to keep doing that all semester. That's all I've got right now. So have a great day. I'll see you later.